Glycolysis. What is glycolysis? Glycolysis is the process in which glucose is broken down to produce energy. It produces two molecules of pyruvate, ATP and ADH, and water. The process takes place in the cytoplasm of a cell and does not require oxygen. It occurs in both aerobic and anaerobic organisms. Glycolysis is the primary step of cellular respiration, which occurs in all organisms. Glycolysis is followed by Krebs cycle. During aerobic respiration, in the absence of oxygen, the cells make small amount of ATP as glycolysis is followed by fermentation. This three metabolic pathway was discovered by three German biochemists. First, Gustav M. Dehn, second, Otto Meyerhoff, and last, Jacob Carol Parnas in the early 19th century, and is known as the EMP pathway, embedding Meyerhoff-Parnas. Glycolysis pathway. Stage 1. A phosphate group is added to glucose in the cell cytoplasm by the action of enzyme hexokinase. In this, a phosphate group is transferred from ATP to glucose, forming glucose 6-phosphate. Stage 2. Glucose 6-phosphate is isomerized into fructose 6-phosphate by the enzyme phosphoglucomatase. Stage 3. The other ATP molecule transfers a phosphate group to fructose 6-phosphate and converts into fructose 1,6-biphosphate by the action of the enzyme phosphofructokinase. Stage 4. The enzyme aldolase converts fructose 1,6-biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which are isomers of each other. Step 5. Triose phosphate isomers converts dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, which is the substrate in the successive step of glycolysis. Step 6. This step undergoes two reactions. The enzyme glyceraldehyde 3-phosphate dehydrogenase transfers one hydrogen molecule from glyceraldehyde phosphate to nicotinamide adenine dinucleotide to form NADH positive H positive. Glyceraldehyde 3-phosphate dehydrogenase add the phosphate to the oxidized glyceraldehyde phosphate to form 1,3-biphosphoglycerate. Step 7. Phosphate is transferred from 1,3-biphosphoglycerate to ADP to form ATP with the help of phosphoglycerokinase. Thus, two molecules of phosphoglycerate and ATP are obtained at the end of this reaction. Step 8. The phosphate of both the phosphoglycerate molecules is relocated from the third to the second carbon to yield two molecules of 2-phosphoglycerate by the enzyme phosphoglycerate-aromatase. Step 9. The enzyme enolase removes a water molecule from 2-phosphoglycerate to form phosphoenol pyruvate. Step 10. A phosphate from phosphoenol pyruvate is transferred to ADP to form pyruvate and ATP by the action of pyruvate kinase. Two molecules of pyruvate and ATP are obtained as the end products. Key points of glycolysis. It is the process in which a glucose molecule is broken down into two molecules of pyruvate. The process takes place in the cytoplasm of plant and animal cells. Six enzymes are involved in the process. The end product of the reaction includes two pyruvate, two ATP, and two NADH molecules. Difference between glycolysis and Krebs cycle. Respiration is an essential process that occurs in living beings in which oxygen is utilized and carbon dioxide is released from the body. The mechanism of cellular respiration involves the following mechanism. Glycolysis, partial breakdown of glucose to pyruvic acid anaerobic. Krebs cycle, complete oxidation of pyruvate to release carbon dioxide, aerobic respiration. Electron transport system, oxidation of NADH and FADH2 to generate ATP. Here, in the article, let us discuss the difference between the Krebs cycle and glycolysis. But first, let us take a look at what each of these terms mean. Glycolysis, it is an anaerobic process which occurs in the cytoplasm of the cell. In glycolysis, partial oxidation of glucose occurs, which yields two molecules of pyruvic acid. Krebs cycle, it is an aerobic process that takes place in the mitochondria of the cell. It gives carbon dioxide after complete oxidation of pyruvic acid formed during glycolysis.